This is a lecture on using different types of analysis to analyze genes involved in aging and longevity, as well as changes in gene expression associated with aging. And we're going to talk a little bit about several different techniques in this lecture, particularly the screening of mutants, genome sequencing, generation of genetically modified organisms, and DNA microarray, and how all of these different techniques can help us study biogerontology. And so, as we've already discussed in the course, model organisms are particularly useful for studying the genetics of aging and longevity. And this is for several reasons that you can see listed here. The first of which is that model organisms tend to have short lifespans, which means we can do experiments in the laboratory in a much shorter amount of time and study aging in something with a much shorter lifespan than a human. Additionally, model organisms have their entire genome sequenced, so we know the locations of every single base pair and every gene in the genomes of these different model organisms like the fly and C. elegans. In addition to having sequenced genomes, there are also readily available mutants in the model organisms that have characterized genotypes as well as characterized phenotypes. And so we can look for genes that modify both aging and longevity in these different, maybe long-lived or short-lived mutants. Additionally, the genes that modulate aging and longevity that have been discovered and studied in model organisms have orthologs, or equivalent genes, in higher organisms that seem to have the same roles. And so there is a lot that we can learn about higher organisms, like mice and humans, by studying the genetics of aging and longevity in these kind of lower organisms, yeast, flies, and worms. And so the first step in sort of identifying some of the genes that are involved in aging and longevity is by conducting what are known as genetic screens. And so even though the sequence of the genome of worms and flies and yeast is known, just having that sequence of nucleotides doesn't really tell us much about the gene's function. And so in order to find out if any of these genes are involved in modifying aging or longevity, we have to start by using a genetic screen, where basically we compare a mutant strain and a normal or wild-type strain and look for differences in aging or longevity between these two different strains of organism. And one thing that makes this slightly difficult is this idea that genes are often pleiotropic. And so when we're lucky, one gene may have one single phenotype, but often they don't, and they're pleiotropic. And one gene actually controls multiple phenotypes. And so in order to successfully conduct a genetic screen and find a good model in which to study aging, you have to find the right mutation to ensure that your gene is in fact affecting longevity in the way that you think it is, and that you're not experiencing any off-target effects. And so if you do find a gene or a mutant with an impact on longevity, we can then identify which genes are involved in either maybe extending longevity or shortening it, or in some type of detrimental effect of aging, by copying that gene or cloning it using PCR. And PCR generates a lot of copies of a very specific piece of DNA. And so you can identify that piece of DNA that you want to copy or clone and multiply it using PCR. And then we can sequence those cloned genes to determine the order of nucleotides. You can do that in several different ways, using several different types of sequencing. And sequencing used to be very time-consuming and very expensive. And so you can see here the cost of sequencing the entire human genome in the early 2000s was almost $100,000. But over the past 15 to 20 years, that cost has decreased dramatically to about $1,000 to sequence the 3 billion base pairs that make up the human genome. And the speed of sequencing has also increased greatly since we moved from what's known as the Sanger method to Illumina sequencing, or what sometimes is called second generation or next generation sequencing. And so if you're curious as to how these different methods work, they're outlined pretty well in the panel in your book. But I am going to talk just for a short minute about Illumina sequencing, just because it is kind of the standard that we use today to sequence large genomes like those of humans. And so there's a machine that does this. And the old way of sequencing, or the Sanger method, was basically sort of like putting together a puzzle of pieces of DNA of various sizes in order to determine the sequence. And that meant it took a really long time. But what Illumina sequencing does is it basically builds the DNA one base at a time and takes a picture after every base is added, so that there's no putting the puzzle together piece by piece. We're actually building the puzzle. And so the way that that works is that you have template DNA on a glass plate inside of this machine, and there are a bunch of nucleotides, A's, T's, C's, and G's, all labeled with different colored fluorophores, or different colors of fluorescence. And in addition, those nucleotides also have this special kind of blocking mechanism that as soon as they're incorporated into the growing strand of DNA, they're blocked from being removed until another compound is added. And so you have your DNA template and then free nucleotides floating around. And so complementary bases will come in and be added one at a time. That fluorophore, whichever base it is, will be glowing, and then the Illumina sequencing machine takes a picture, records the color of that fluorophore, and then the machine washes away both the fluorophore and this block, leaving the nucleotide behind. And the next nucleotide that's complementary to the sequence of the template can be added. And so it's a series of incorporating a nucleotide, blocking it long enough for the sequencing machine to take a picture, note whether it's an A, T, C, or G at that position, wash off the fluorophore in the block, and on to the next nucleotide. And so this process has greatly reduced how long it takes to piece together a sequence, because rather than, like I said, piecing it together from a lot of different size pieces, we actually build it one step at a time. And the machine takes into account each base in order as the DNA is made. And so after you've screened through mutants, and they definitely have impacts on longevity, you've cloned the gene, you've sequenced it, you then can make some predictions about, one, the sequence of amino acids, as well as the structure of that particular protein encoded by the gene. And so there are programs that can be used to basically translate the sequence of amino acids determined by, or the sequence of amino acids in this gene. And from that primary structure, you can actually determine some of the function, because primary structure, as you noted in this previous lecture, has the instructions for folding of the protein. And folding, or specific folds, can dictate specific functions in a protein. And so just by knowing the order of amino acids, you can start to predict what that gene's function is and what it does. And so we've made it from an observed phenotype to the protein involved within our model organism. And that is a process known as forward genetics, where we knew the phenotype. Maybe it was something detrimental that happens during aging we were interested in studying. Maybe it was something that seemed to extend lifespan. Maybe lifespan was shortened. And we used forward genetics, i.e. PCR cloning of the gene as well as sequencing, to figure out which gene underlies that age-related phenotype, or that longevity phenotype. And as I said before, this is known as forward genetics, and it's how we begin to identify and understand the genes involved in aging and longevity.